Could an injection of your own brain cells be a way to halt multiple sclerosis, or MS? It sounds surprising, but that's what researchers in the US are finding in mice with a rodent version of the disease. In MS, the immune system attacks the brain and spinal cord, damaging a material called myelin that wraps around and supports nerve fibres. But an extract of this myelin material, made from the oligodendrocyte brain cells that normally produce it, if injected into the bloodstream, can permanently deactivate the rogue immune cells responsible for causing MS to happen. I'm Chris Smith, and Abdol Mohamed Rostami is one of the researchers behind the discovery, and he told me what they're doing.
The current therapies for MS relies on suppressing the immune response. The problem is that we make patients prone to infection and sometimes to cancer. So in this study, we try to find a way to suppress the immune response only to the brain and leave the rest of the immune system intact. So we have an animal model of MS, which mimics all the clinical features and pathology of multiple sclerosis. And when we inject a protein from the brain into the vein of these animals, which can cause suppression of disease in a way that animals can walk again. These animals were paralyzed before, but we injected this protein IV, they could walk again.
When you say you injected a protein from the brain, what was it you were injecting into the animals?
We injected part of the membrane of a cell in the brain named oligodendrocytes. These cells produce myelin sheath around the axons in the brain. So we produced tiny sacs from these membranes and injected those into the vein of these animals.
This is extraordinary because in a person with multiple sclerosis, the thing that's being attacked by the immune system are those very cells, those oligodendrocytes, and specifically components of the myelin that they wrap around nerve cells. And you're saying you're taking bits of that and putting it into the bloodstream, and it seems to make these animals with the mouse equivalent of MS decidedly better. So what on earth is going on then?
You're right. We induce something called immunological tolerance. We try to educate the immune cells not to attack that tissue that we are injecting into the bloodstream. What happens, those parts of the immune system attack the brain, they die, they go away. Therefore, they're not there to attack the brain anymore.
How long does the therapeutic effect persist for, or is it indefinite? So you treat them once, you get lifelong benefit, or do you have to keep doing this to keep the disease under control?
In the experimental animal, even one dose was enough to keep the effect till the end of the animal's life.
And were there any other consequential effects, in addition to benefiting them from their MS-like condition? Were there any other side effects or negative effects?
No, we actually looked at the immune system of the animals. They are not suppressed. That's the beauty of this technique. Unlike other therapies, which weakens the immune response, the immune response of these mice are basically intact, except for those cells which are reactive to the brain and damage the brain, which is removed or weakened. The rest of the immune system is intact.
Obviously, these are mice that you're working on. They are not humans. They have quite a different brain structure. They live for a different length of time, and the disease is an artificial one that you create in these mice. So to what extent do you think you can actually extrapolate this to humans?
Yeah, most therapies we have in MS right now have been tried in this model first. So we have confidence that this model could be used to try therapies in the experimental model first, then try it in human disease.
And is that the idea, that you're now going to try and translate this to patients who have got a diagnosis of MS to see if you can gain control of their disease in the same way?
Yes, exactly. We're trying to see if we prepare vesicles from human oligodendrocytes and inject to the same patient, we can educate the immune system so they don't attack the brain, and at the same time leave the rest of the immune system intact.
Encouraging stuff, isn't it? Abdol Mohamed Rostami, he's at Thomas Jefferson University.